Our next speaker is Susan Horwitz. Now, Susan's going to talk about all of the patients. Well, I think it's 1 million patients, is that right? The Taxol. Susan and Taxol, and what she has done in appreciating that from natural products there can be a pharmaceutical that has great benefit. And she's been working on this for many, many years, and she recently she has support from the Housewives Charity Foundation. Now this is one is that the name?
That's my neighbors.
Is it Housewives Charity Foundation? It says that the support
comes through the Breast Cancer Research Foundation.
Okay.
I have support from the Breast Cancer Research Foundation.
It just shows you there are foundations and there are foundations to support important research endeavors. So Susan, come up and tell us how this is all going and the work that you are continuing to do, and with this huge number of patients, exactly what you're finding.
Thank you very much. Not easy to follow BioX, I must say. I'm a working scientist. I have a laboratory at the Albert Einstein College of Medicine, and I'm in the lab almost every day, almost. So I didn't develop this 40 years in a lab. 1 million Taxol patients later. This must have come from you. So it's a sort of interesting title that I never would have picked myself, but it's interesting. So that's what I'm going to tell you about. As you can probably see, this is the branch of a tree, and it's from the bark of this tree that two medicinal chemists, Monroe Wall and Mansuwani, actually isolated in the 1960s the molecule Taxol. And it was a very difficult job, and they deserve a great deal of credit for it. It's a complex molecule, and it's only present in 0.001% by weight from this bark. And actually, they even got the structure right, which, as I'll show you later, is extraordinarily complex. And you have to remember that in the 1960s, chemistry was quite different from what it is today. So I thought that I would tell you today how I got into doing this work, and a really rather personal story about myself. And this was nothing that I had ever planned to do. I was a graduate student writing my thesis on basically new enzymes that I had isolated from bacteria, studied many of their characteristics, and I became very interested in enzyme kinetics. And I had planned on going on and doing a postdoc in that area. But 5 days after I defended my thesis, I gave birth to twins. So my life was quite different than I had expected. My husband was an intern at the time, working every other night, making $1,200 a year. That's what interns were making in those days. I wanted to work. I loved science, and frankly, I needed the money. And I went to speak to my potential postdoc mentor, and I said to him, do you think I could work 3 days a week, Tuesdays, Wednesdays, and Thursdays? And he looked at me like I had dropped from another planet, and that was the end of enzyme kinetics. So I went back to my mentor and I said, this is what I want to do. Do you think that it would be possible? And he had a close friend who was chairman of the Department of Pharmacology at Tufts Medical School. And he said, I'll take her 3 days a week. But the deal was that I had to teach pharmacology, one section of pharmacology, to the dental students. Frankly, you have to remember that at this time, pharmacology was a totally different study than it is today. It was really sort of drums and taking blood pressure of dogs and cats. Nothing like we think of today. We talk about molecular pharmacology. But I looked at the various areas and I said, well, I'll give the lectures on anti-tumor agents because that was sort of the most biochemical aspect of pharmacology, and that's what I did. So To make a long story short, I loved it. And it was a wonderful thing. I love the idea that small molecules, by that I mean 500 molecular weight or so, can really do great things. You have a headache, you take an aspirin, the headache goes away. And I decided that this is really something that I wanted to study, and I became particularly interested in molecules that could be isolated from natural products. So some of us forget that up until very recently, most of medicine was natural products, these types of drugs, and they come from really what I call natural products. All of us know about aspirin. Every day I hear something new about aspirin. I just heard on the television, if you take aspirin, your chances of getting liver cancer are reduced. An amazing small molecule. Then, of course, quinine has been used in malaria and morphine for pain. How important steroids in the pill have been, which originated from the Mexican yam, changed the world, basically. Penicillin, during the Second World War, was an amazing drug and continues to be. How many of us take one of these cholesterol-lowering drugs? And I've added Taxol on my own that comes from the yew tree, or as you could see on that first slide, the botanical name, Taxus brevifolia. So this is really what I spent my scientific life looking at small molecules from natural products that could be useful in the treatment, particularly of cancer. So how did I ever start this? After I spent my 2 years at Tufts Medical School, I then spent 2 years working 3 days a week at Emory Medical School. My husband went into the public health service, and then I came to Albert Einstein and I worked there 3 days a week. At which point I was making $4,500 a year, barely enough to pay my babysitter, and my children were going to the first grade, and I decided that it was now or never, and that I was going to go back full-time, and so I made that decision. I had already worked with a number of molecules while I was a postdoc in all these different places. I had done some of the earliest work on the camptothecins, on the epipodophyllotoxins, and I was very much involved in bleomycin at the time, when I was very fortunate to be asked if I would be an instructor at Albert Einstein by the chairman of pharmacology, who made it pretty clear that I would be an instructor forever. But I have to say, to his credit, within 2 years he came to me and said, I want to make you an assistant professor. So I received this letter from the National Cancer Institute, and those of you who work with the NCI know that you just don't get letters like this anymore that say, would you please study this compound in your systems? Of course, they didn't give me any money, even then, but they did write me this letter. And it turns out the compound has absolutely nothing to do with protein synthesis, but the compound is incredibly insoluble, very hydrophobic, and about that they were correct. So what they sent me, the folder enclosed, was the single paper that was published by Monroe Wall and Mansukhwani and their colleagues in 1971 in the Journal of the American Chemical Society, which is now truly a landmark paper in which they discussed how they purified it. They purified it every step of the way by taking the material and seeing if it would kill cells. That's really a tedious way to work, but it was the only way that was available to them at the time. And I looked at this, and I had a new graduate student, Peter Schiff. I said, what do you think? Well, the thing that encouraged me to go do this was the structure. And I know that you aren't all chemists, but believe me when I tell you, this is truly, to me, a beautiful structure. And I say that this is the kind of a structure that only a tree would make. No chemist would sit down in their laboratory and make this drug. And I'm not going to go through all the things that make it so special, but just one little thing, for an example, this 4-membered octane ring out there and here. It's the kind of thing you just never see, and it's very difficult to put this kind of thing together. So I wrote back to the NCI and I said to them, please send me 10 milligrams of the drug and we will look at it and see what we can come up with. basically, at this point, they had the molecule, a small amount of it, and they knew that it killed cells. And they also knew that at the NCI they had tested this and it actually had reduced the growth of some tumors in mice. But nobody knew how it was doing it. And that was what they asked me to do. So let me tell you just a little bit about this tree. And I could give a whole lecture on the tree. It's a fascinating story, this Western yew tree, and also how it's been used in literature. It's really very unusual. So I work in the Bronx at the Albert Einstein College of Medicine. It's very close to the New York Botanical Gardens. And as soon as I got this letter, when we decided to get the drug, I went over to the Botanical Garden and asked them about the tree, and et cetera. And they said, well, there are 2 very large trees in Greenwich, Connecticut. I said, oh, wonderful, that's very close. And they said, they told me it's on the main street at the church, and I went there. I couldn't find the trees, so I asked at the church, and they said, oh, yes, they're in the cemetery in the back of the tree, in the back of the church. So we went to look in the back of the church, and sure enough, these are magnificent yew trees, Taxus brevifolia, among the tombstones, as you can see. If you go to the British Isles now, you will see this all over, these yew trees that are in the cemeteries. And the reason is that if you look in the literature and you read about this tree, it was believed that these trees really were associated with death, partly because the wood from these trees was used by Robin Hood for the bows and arrows that the English used, and that made it very, very valuable. Also, there was a folklore about these trees, and it was believed that the roots of these trees could speak to the dead and reveal their secrets. So here I took this picture of this, and I just give you a couple of literary things. As I said, I give a whole lecture on this tree and all. But as you can see, this was by T.S. Eliot. It was used by Shakespeare. Sylvia Plath used this, and you can see. But when the voice is shaken from the yew tree drift away, let the other yew be shaken in reply. I always think of this because I struggled through T.S. Eliot when I was a freshman in college, never dreaming that I would use something like this. But when I told my grandchildren about this, they insisted that I include this. So I'm sorry to say I sold the wand that did it. 13.5 inches yew, powerful wand. I'll tell you a little aside story. Recently I was in Germany. I gave a lecture, and I mentioned this, and I said my grandchildren suggested this. And a young woman who was an assistant professor came up to me afterwards. I think she was from Birmingham, and she said, Dr. Horwitz, I loved your lecture. It's the first lecture I ever heard from a grandmother. And I thought to myself, the world is changing. You better get used to this. So when we got the drug into the lab, we got the 10 milligrams. What do you do? This is a drug. It's obviously toxic. It's killing cells. What do you do? So the first thing we tried to do was ask the question, does this drug have any effect in a specific part of the cell cycle? And you can see here that you have cells that have a diploid and tetraploid amount of DNA. These cells with the Taxol go through a perfectly normal S phase. They're not affecting DNA synthesis, but all the cells are blocked here. They don't divide. So this was of interest to us, and we then took those cells and looked at them under the microscope. And what we saw that these cells were absolutely filled with microtubules, which are an important organelle in the cell, which allow the cell to move. But most importantly, microtubules are important because they give you a normal cell division with 2 daughter cells with equal amounts of DNA. So when I looked under that microscope and saw those cells, that's sort of a eureka moment for an assistant professor who's looking for a niche and says, gee, this is something nobody else has seen, and it was very exciting. So this is what a microtubule looks like. It's made up of an alpha and beta tubulin. You form these heterodimers, and then you form protofilaments, which go to make a hollow tube, which is a microtubule. And the important thing is that this microtubule is very dynamic. It has to be able to shrink and grow, and in order to do the many things it has to do in a cell. And what I'll show you is that the drug Taxol has a specific binding site inside the lumen of this microtubule, and that it binds to the beta tubulin, and when it does that, it paralyzes the cytoskeleton of the cell, so that the cell cannot behave normally, it cannot divide, and therefore this drug inhibits the ability of cells to divide. So this shows you a cell here. The blue is the DNA, the nucleus. The green are the microtubules. These microtubules, as I said, divide the cell, divide the DNA, but they're also actually used for little organelles to run over move around in the cell. And when you treat with Taxol, you can see how completely different the microtubule cytoskeleton is, and so that cell cannot divide. And if you see a patient who's taking Taxol and you look at their white blood cells, you see all these bundles of microtubules being present. So you can also study this in a test tube by isolating tubulin from the brain. It's soluble, and you can watch it make a microtubule in this fashion here. This is a normal microtubule being made, and in the presence of GTP. And then when you add calcium or you make it cold, it depolymerizes. In the presence of Taxol, you see you polymerize this microtubule, and it's absolutely stable. It won't depolymerize. And in fact, if you take a normal microtubule and you add Taxol, it instantly stabilizes it. So we had a drug that we knew had something to do with microtubules, clearly. We knew the microtubules were stable, but how were we going to find out where this drug binds in the microtubule and actually what it does to the microtubule? And that's hard to do because this doesn't make a covalent bond, and so we thought if we could get some kind of a label on the drug, it would be helpful for us. At that time, I was invited to Hawaii to a meeting to speak about bleomycin, which I was deeply involved with at the time. And at that meeting, I met John Duros, who was the head of new drug development at the National Cancer Institute. And I was very excited about what we were doing. This was a wonderful small molecule. When I started to work with this, nobody was interested in what I was doing. And fortunately, the times were easier. I was able to get both an ACS and an NIH grant, but with very little preliminary results. And as I say, there was only that single paper in the literature. By this time, I realized that this drug would be a fabulous tool for many scientists, but of course I had no idea that it might be a useful agent with patients. So I told him all about what I was doing, and he said to me, Susan, when you return back to New York, you should write me a letter and tell me what you told me and what you need. So I wrote to him and I said, although we've not completely defined its site of action, we know that it's quite different from any other drug we've previously studied, and we plan to pursue it and study it. And I thought maybe he would be able to get us some labeled drug. I never heard from him. I never got an answer to this letter. 25 years later, when Monroe Wall was actually ready to retire, and he was cleaning out all his files, he wrote to me and he said, Susan, did you ever see this letter? It was written, as you can see, right after I wrote to him, and it said, Dear Monroe, can you help this poor girl? Enclosed letter. Well, I'm happy I didn't see it for 25 years, because by that time I was no longer a poor girl and I didn't have to worry about him. But it's, of course, took place in the 70s, but it's a letter that hopefully nobody will write today, particularly on NCI stationery. Well, we never got any drug from them, obviously. But we had gone on and we made our own drug. That's the bottom line to so many things. Do it yourself. You wait for other people, you never get it. So this jumps forward a great deal, of course, to many studies that we did, but lots of other people. Even Argolis was very important in this study, doing actually electron microscopy. But the bottom line is this, that Taxol has a very specific binding site in beta tubulin. I can show you right here. And the presence of that molecule alters the interaction of this part of the molecule, the M loop, and this part, which goes together to make that protofilament that I showed you. And it's quite interesting because in the alpha tubulin, which Taxol does not interact with, there's a much larger loop here, which has 7 more amino acids, which is missing in the beta tubulin. So there's a place in the beta tubulin for this molecule to move into, which isn't present in the alpha tubulin, and which allows the drug to stabilize the microtubules. So I want to tell you a little bit about the chronology of this drug and how it came about and what we did. I am not a physician. I do not treat patients, but I'm very interested in the development of these drugs. So I'm not going to go back all the way. I often say sometimes when I show this to students, most of you weren't born then, but I have to say that to everybody today, but to some of you weren't born. But this was really a collaboration between the Cancer Institute and the Department of Agriculture, who got together to do these absolutely random collections. And presumably Taxus brevifolia was really the last collection. They took students and sent them out to California and Oregon during the summer, and this presumably was at the end of August. And as I told you, this was the original paper by Monroe Wall and Mansukwani, and they published the structure. But there's nothing special here about Taxol. There are many, many compounds in those days going through the National Cancer Institute, and really nothing special about these activities. They were positive, but they weren't outstanding. In '79, we published our first paper on the mechanism of action of this drug. And I think the fact that we were able to show that this was a drug that worked in a new way, other than any other drugs that were then being used for patients, was very important. And I say this because I think it's so important that students who are in biomedical education realize that the work that they are doing in the laboratory really can be used so important in translational medicine and in the development of drugs and the treatment of patients. So that's, I think, is a wonderful example of how that happened. And then when they started to do preclinical toxicology, as I told you, this drug is extraordinarily insoluble, which has presented formulation problems continuously. It also had neutropenia. It dropped white blood cells in the patients, but it was still approved for a new drug application. In '83, when the clinical trial started, the first patient that got Taxol died of an anaphylactic shock. That's an allergic reaction. And so as you can see here, there were 5 years when actually people had to get together, particularly oncologists, clinicians, pharmacologists, to really develop ways that this drug could be used. Again, many drugs would have been dropped after the first patient died. But we really pushed this. And I think very important in drug development is that each drug has an advocate, someone who really believes in the drug and is willing to speak up about that. There was also, of course, a tremendous scarcity of the drug because it was being isolated from the bark of a tree. But what happened during those 5 years that we learned that You had to pre-treat patients with steroids and antihistamines. You had to give the drug over a period of time instead of, as done was originally, as a bolus altogether. And all of those things made a tremendous difference. And then when it went back in clinical trials, you can see that right away it showed clinical activity, and particularly drug-refractory ovarian cancer, metastatic breast, and it's been used in lung and many other kinds of cancers, and it's continually being evaluated with new drugs. And now in the era of personalized medicine, when new drugs are coming out, Taxol is very often used in collaboration with these new drugs. But then this was a very unusual situation, because this was happening in the 60s and 70s, and nobody patented anything. Today, people patent their sneezes, not then. So here was this drug, which clearly had clinical activity, and how do you move forward with this? Because the National Cancer Institute is not a organization to develop drugs. So actually, the NCI put out something called a CRADA, Cooperative Research and Development Agreement, and various drug companies bid for this. It went to Bristol Myers Squibb, and for their ability to put the drug out on the market, they would receive 5 years of exclusivity. So that went in '91, and they did a very good job of moving the drug forward. So it was first approved for ovarian cancer in '92, and then for many, many other cancers. So how do we get Taxol today? Because all of this started coming from the bark. So what Bristol Myers Squibb was able to do was to actually isolate part of the molecule. I should have showed you this when I was doing it. No. From the, well, this isn't working. Anyways. What happened to it? What we do today is that we actually, from the needles of the tree, we isolate the complex ring system. And Bob Holton, actually at the University of Florida in Gainesville, was responsible for the Florida State, pardon me. It's good. It was worth a bundle, which he didn't spend very well, but that's beside the point. Anyways, you can isolate from the needles the complex part of the molecule, which is very difficult to synthesize, and then put on the side chain. And so about 50% of the Taxol used today comes from that, but about 50% comes from plant tissue culture. Cells from the needles which are grown in tissue culture and produce the drug. So this is really quite interesting, and that's how the drug comes today. Okay, let's see if I can go on. Okay, so many things happened with this drug that I wasn't prepared for, that I didn't think would happen, but it became a whole question of public-private partnerships, because that's what happened between Bristol Myers Squibb and the National Cancer Institute. And then there were a whole series of congressional hearings which focused on the procedure by which BMS had been chosen as the Taxol CRADA partner, and was the exclusivity that was given to them in the best interest of the citizens of the United States. And then the question, the major issue, is who decides on the price of Taxol? After all, it was the United States government that collected all this bark. It were people in academia who did so much of the work, and then Bristol Myers Squibb was actually putting it on the market, and who decides on the price. These are very intricate and complicated problems. But really, who pays for medical research in this country? It's mainly us, our taxes, through the NCI, and of course also from philanthropy. So this brought me into areas I never dreamt that I would be thinking about when I was a student. In addition, Taxol was the name of the drug given to it by Monroe Wall from Taxus brevifolia. It was an alcohol. And all of a sudden, Bristol said, that's our name. Somehow or other, they got it as a trademark. And I was told, as were other people, that I wasn't to use this word anymore, that this was now the trademark, and the generic name was paclitaxel. So people often ask me, what's the difference between Taxol and paclitaxel? There is no difference. They're exactly the same. And a very important drug, Taxotere, which was is a derivative of Taxol that is made in the laboratory, also had that trademark, and the generic name is doxytaxel. So there's many, many aspects of this drug development that I was never aware of at the time. And there are many other questions that came up at this time having to do with natural products and the environmental policy. So, I mean, what does a pharmaceutical company owe a country, you know, where they got a drug from? In this instance, it was from the United States. But over the years, many drug companies went into countries, Costa Rica, Panama, actually, you know, took out things, isolated drugs. What do you owe those countries where you took it from? And more drugs now are coming from the sea, because that is an area that wasn't exploited earlier, sponges and corals. So there were many competing factions, and I was extraordinarily naive about all of these things. But for an example, the Environmental Defense Fund wanted me to speak for them, and they wanted to list Taxol as an endangered species. At first I thought it was a great idea, but then I said, we won't have the tree. There were people who were very concerned about the loss of the old-growth forests in the Northwest. That's where the very special spotted owl lived, if we were going to cut down all the trees. And sort of the media got involved in pitting the environmentalists against cancer patients. And also it brought up, of course, a very important question, that of preserving ecosystems and biodiversity in all areas, because undoubtedly there are other Taxols out there that we don't know about and that we haven't had the opportunity to look for. So when I first started working with Taxol, I said this must be a prototype for a new kind of class of drugs. But there was nothing found beside Taxol for 15 years. At that point, it became clear that Taxol was going to make money, and then other people started, of course, looking. Oh, and another question that became very important was who gets the drug. In '87, Bill McGuire, who was at Hopkins at the time, said 2 of my 12 patients have shown dramatic improvement. That was very major in refractory ovarian cancer. And ASCO that year, 30% response rate in ovarian cancer was reported. When you have a drug that's very scarce, how do you decide who gets the drug? I remember that there was a judge at that time, Supreme Court judge, whose wife had ovarian cancer. He called the president. I mean, it's very, I never realized how complicated these things are when you have a scarce drug. I went to visit Weyerhaeuser in '88 with some people from the NCI to discuss with them the mass propagation of taxes from seedlings and production of Taxol by tissue culture. And then there was a very important ethical question. When there's a shortage, who gets the drug and who makes the decisions? So all of these things were obviously things that I had no intention of thinking about when I started in the business, but they've all added certainly to my depth and understanding of drug development. So why is this drug so important? It was certainly the major anti-tumor drug that was developed in the 1990s. It is used a great deal today, as I say, even with these new very specific personalized drugs that are being developed. And the commercial product really resulted from a collaboration of the government, basic and clinical investigators, academia, and industry. And as we heard Carla say, it's a collaboration which has really put this together. And it became a blockbuster drug. Bristol made $2 billion a year from it, but I think this was really the drug that spurred biotech and pharma to be interested in cancer drugs because they realized that they could really make money from this. So I have a little more to tell you. So after these 15 years, it became clear that there would be money to be made, and as I said, people started looking. And the interesting thing is that I put here 5 different living organisms that all produce a molecule which acts like Taxol, but has a totally different structure. So it's like an evolution to this particular mechanism. But here you see, of course, the tree. These are the tree, and this is, of course, the drug structure that you already know. But there are other drugs now which have become FDA approved. So for this is a bacteria here, Myxobacterium, and from it came from a soil sample in Africa, and it was isolated in East Germany. And you can see a structure below it, the epithylones, and one of these, ixabepilone, is now approved, FDA approved, for metastatic breast. I'm particularly interested in this molecule, discodermalide, which comes from a sponge, has a totally different structure, but also interacts with microtubules and stabilizes them. And then 2 other drugs here from sponges, which actually come from New Zealand. And you can see these structures are different. They all stabilize microtubules. They all bind to beta-tubulin, but these 3 bind to one site of beta-tubulin, and those 2 bind to another site. And as we're studying more, we realize that even though these bind at the same site, they do not make exactly the same interactions with this hydrophobic site. And I just want to end by showing you that. We have actually developed, loads of people do hydrogen deuterium exchange and mass spectrometry, but we have actually developed this to be able to use this with microtubules and to be able to look at some of the allosteric effects of the drug. So we know where the drug binds, but what does the presence of the drug do to the whole microtubule? And by using hydrogen deuterium exchange and mass spectrometry, we've been able to actually look at that. And I have some data here, but I'm not going to go through it, just to show you the very end. We're interested in the ability of discodermalide and Taxol to be synergistic, which this is in a xenograph in a mouse, and you can see that together these 2 drugs decrease the size of the tumor, and long as it's given, keep the tumor non-existent. And we want to understand how this synergy works. So we've been using this method, and we exchange the deuterium, and then we can look to see that both drugs actually decrease the deuterium, but we can't get anything specific how these are different. But what we've now done is actually chop up the microtubule and look at every little peptide and compare these peptides, normal, Taxol, and discodermalide. And when we do this, what we found that there are differences between these 2 drugs that can help us to understand why these 2 drugs, when they're given together, have better results in reducing tumor size. I won't go through this. But in the end, what we've been able to do is model how these 2 drugs go into the microtubule. And what you can see here, the Taxol is in red and the discodermalide is in blue, is that they do come together in this binding site, but then they're different. And the Taxol moves in one direction and the discodermalide moves in another direction. So I have been collaborating on this discodermalide project with Professor Amos Smith from the University of Pennsylvania, a very good chemist. And together we thought, wouldn't it be great if we could take this information and actually maybe synthesize some molecules that in themselves may have better activity. And so you can see here the two structures. This is Taxol, this could journalize, and now we have made a whole series of hybrids from two natural products. And I can't give you the mouse data because we don't have it yet, but some of these hybrid molecules are better in tissue culture than either of these drugs alone. So I'm going to end here. Well, the conclusion is this is a good technique to use, and we plan to use it. These are, of course, I don't do this myself. I just want to point out two wonderful students, Marina Kropanavar-Bain, who's been an MD PhD student in my lab and set up all the mass spec together with Hui Chia. So, starting with a very simple story of how we had this molecule, how we began to study it, and nobody really cared about what we were doing at the time. I thought of it as a good, certainly a tool for biochemists interested in the cytoskeleton. I never dreamt at that time that this drug would have been given to over 1 million patients as it has been now. And now, using this information that we're getting from natural products, we're trying to synthesize new molecules that may be more helpful in the treatment of cancer. So that's my story. And I'm happy to answer any questions if you have any. Yes.
I have two questions. It's a wonderful story. Thank you. One, what are the plants doing with these very strange chemicals? And secondly, other than your own persistence and fortitude, what were the conditions or the people that made your career possible?
I'll answer the first question last, but this is in my mind. Certainly, the people who were taking me on at 3 days a week, they were, I wouldn't be here if it weren't for them. Because I couldn't work 5 days a week with 2 twins and a husband who's rarely home, and I don't have any knowledge. It would have been impossible. So, yes, it was the late Dr. Maurice Friedkin, who was chairman of TUS at that point. And I'm very grateful to him for allowing that. And then he had a friend at Emory who took me on when I went there. And then I think at Einstein, and I had a friend there who took me on. Without those people, I could not have done it. No question. So for me, I mean, I really feel strongly that women, or men for that matter, don't have to do everything exactly the same way. We don't all have to get our PhD and then take a postdoc and then do this and do that. There are many ways to have a wonderful, rewarding career in your own time. Sure, I would have been a full professor earlier if I had done the other things, but I have no regrets whatsoever for the way I did. And the other question was this, oh, okay. So, you know, that's an interesting question, and we don't know everything about that. But certainly, I showed you 5 things, living things on that slide, and not one of those living things is capable of moving very much. A tree cannot move. Corals and sponges are attached to rocks. And yet these, particularly Taxol trees, which have evolved over many, many centuries, and they've lived. And the reason is, I think, obviously, it's not an original idea. They have evolved and made these compounds in their body which protects them, so that they are protected from a variety of insects, birds, whatever would kill them. Because most of these organisms which give us these drugs are ancient, and they've been around for a very long time. And they have evolved these drugs. So Taxol, for an example, many chemists have tried to modify it to make it more water-soluble, to do this, that. But it's very good the way it is. And you can't do much to it. Other drugs like aspirin, although they do come from the willow tree,
they have been modified over many different ways. So they're both ways. But what really these metabolites do in the tree, we really don't know. But all of these living organisms, not the bacteria, but certainly the tree, have microtubules. And I haven't been able to look at microtubules from these, but we did at one point look at microtubules from plants, and they certainly interact with Taxol. So somehow these living organisms are able to separate these toxic molecules they make from their own receptors. And how that happens, I don't know. But it's fascinating. Unfortunately, there's not a lot of money for people to get grants to look at these sorts of things in plants. For the most part, they're just not that interesting. People, the National Institute is in that problem. Yes.
Really interesting story. I have a question because I heard that a lot of companies are trying to do microtubular target drugs, and some of them are working better than Taxol, but none of them worked. So there are discussions about Taxol's definite targets microtubular, and there may be additional target in the cells that we don't know about. I just wonder what your comment is.
Okay, two things. Certainly, the question always arises, are there off-target results?
Not just off target, but there could be additional target.
I don't deny that.
Yeah.
But I haven't found any. And we have looked. But that doesn't mean that they're not there. By any means. There are things out that BCL2, which is a death protein, does have a binding site for Taxol, and I believe that. I haven't studied it, but I believe that definitely. There is a very interesting drug called Abraxane, which some of you may have heard of, which is Taxol as a nanoparticle with serum albumin. One of the big areas in pharmacology today is formulating drugs in different ways. And this drug, the company is Abraxane, and that drug doesn't keep people alive longer than a metastatic breast cancer, but it reduces the side effects. I mean, I haven't talked about side effects, but all of these molecules have side effects. And one of the side effects of Taxol is neurotoxicity, not extremity toxicity. So in the hands and the legs, people have terrible problems with toxicity because there's a lot of microtubules in the brain and other places. And I think that Abraxane as a nanoparticle, serum albumin, reduces that. It also reduces the loss of hair. Women who are taking Taxol lose every hair on their body. So these kinds of things in formulation are very, very important. And there's a lot of interesting work in that area.
Just a comment first. Thank you for a very interesting, wonderful story.
Thank you.
Science. There's so many stories within your talk, but I just wanted to note that in 1992, when this drug was, you mentioned it was submitted as a new drug application in 1992 in July, and was approved in December of that year, which is unprecedented. The fact that it got approved so quickly speaks to how desperately it was needed at that time when there was so little available for treating cancer patients.
Well, that's why I said that when this came out with ovarian cancer, everybody wanted the drug. And that product, many ethical and problems gets it when you don't have enough of it to go. Thank you very much.
Good for your story.
We are going to have lunch downstairs on 9. We will try and stay on schedule, so if you can get down there, get food, and we'll have a presentation at lunch as well. There are restrooms on both floors. There's a glass stairway that goes down to 9 and will connect us down there. And we will, following lunch, we will be back here at 1:30 to continue, and hopefully you'll have some time to at least talk to each other a little bit over lunch, as well as a great talk. Thanks.